Computer aided drug designing methods, second video about bioinformatics topic. So let's try it. Computer aided drug design uses computational chemistry to discover, enhance, or study drugs and related biological active molecules. The most fundamental goal is to predict whether a given molecule will bind to a target, and if so, how strongly. So molecular mechanics or molecular dynamics are most often used to predict the conformation of the small molecule and to model conformational changes in the biological target that may occur when the small molecule binds to it. So semi-empirical ab initio quantum chemistry methods or density functional theory are often used to provide optimized parameters for the molecular mechanics calculations and also provide an estimate of the electronic properties, electrostatic potential, polarizability, etc. of the drug candidate that will influence binding affinity. So molecular mechanics methods may also be used to provide semi-quantitative prediction of the binding affinity. Also knowledge-based scoring function may be used to provide binding affinity estimates. These methods use linear regression, machine learning, natural nets or other statistical techniques to derive predictive binding affinity equations by fitting experimental affinities to computationally derived interaction energies between the small molecule and the target. So ideally, the computational method should be able to predict affinity between a compound synthesized, and hence in theory only one compound needs to be synthesized. The reality, however, is that present computational methods are imperfect and provide at best only quantitatively accurate estimates of affinity. Therefore, in practice, it still takes several iterations of design, synthesis, and testing before an optimal molecule is discovered. On the other hand, computational methods have accelerated discovery by reducing the number of iterations required and in addition have often provided more novel small molecular structures. So drug design with the help of computers may be used at any of the following stages of drug discovery, like hit identification using virtual screening, like structure or ligand based design, hit to lead optimization of affinity and selectivity, then structure based design, QSAR, etc., lead optimization of other pharmaceutical properties while maintaining affinity. Then de novo method. De novo design is the approach to build a customized ligand for a given receptor. This approach involves the ligand optimization. Ligand optimization can be done by analyzing protein active site properties that could be probable area of contact by the ligand. The analyzed active site properties are described to negative image of proteins such as hydrogen bonded donor, hydrogen bonded acceptor, and hydrophobic contact region. There are two types of de novo design, like inside out and outside in. So de novo design is the approach to build a customized ligand for a given receptor. This is the de novo. Novo means something new. So this approach involves the ligand optimization. Ligand optimization can be done by analyzing protein active site properties that could be probable area of contact by the ligand. The analyzed active site properties are described negative image of proteins such as hydrogen bond donor, hydrogen bond acceptor, and hydrophobic contact region. There are two types of de novo design, like inside out and outside in. In each of the methods, you need to follow these steps. Identify points on the receptor where a ligand can form favorable interactions. Choose ligand groups of atoms that are complementary to these sites. Then connect them into a molecular framework in an exhaustive, unbiased way. Then ADEME tox property prediction. Drug discovery needs the safety of what the drug does on the body during curing a disease without causing harmful side effects and of what the body does on the drug, namely ADME tox. ADME tox. Absorption, distribution, metabolism, excretion, and toxicity of the drug. The most challenging aspect of drug design and development is the rule of three relating to activity exposure toxicity. So ADME tox properties are widely used in drug discovery to optimize this balance of properties necessary to convert lead optimization compounds into drugs that are safe and effective for human patients. The best approach towards predicting ADME tox properties in early stages of drug discovery involved the evaluation of drugs as lead compounds and may be done simultaneously while activity assays are being carried out. It would be desirable to predict the correlation between the ADME tox properties and the chemical structure in the course of designing a drug. So failures in the drug discovery process account for a substantial part of the cost because many compounds are discarded after heavy investment owing to unforeseen toxicological effects. It is therefore suggested that the ADMET tox property assessment is done at the hit identification stage. Then hit identification. Target identification and edification is followed by hit identification, where the application of ADMET begins. Thousands of chemical compounds are screened from compound libraries through high throughput biological assays and subsequently subjected into in silico ADMET models for identification of compounds that are acceptable for drug-like properties and the rest discarded. The purpose of ADMET application at this stage is to identify potential weakness and liabilities in the selected compounds to focus on improvement or optimization efforts. Then lead identification stage. After hit confirmation, a series of compounds is selected for their drug-like qualities and subjected to applications of in vitro and in silico technologies. The in silico model focuses on predictions of chemical modifications to improve ADMET properties. Mechanistic models are considered appropriate as predictions based on atomic and molecular interactions are more accurate. However, in order to focus on measurement of ADMET properties of newly synthesized compounds, in vitro and assays are simultaneously carried out. Then lead optimization. Lead optimization is concerned with experimental testing and confirmation of the compound based on animal efficacy models and ADMET, in vitro and in situ tools. So application of ADMET tox tools has increased the success rate of drug development as well as helped in reducing the cost and time factors. The use of in silico and in vitro ADMET tox has found universal acceptance. In silico tools provide a much higher throughput, but they suffer from adequate predictability which limits their use. However, the reliance on in silico models is due to their ability to predict which compounds should be synthesized based on confirmed hits and structural modifications, since it helps in selecting a drug-like compound. The best way to implement the ADMET tox property prediction is the integration of both in silico and in vitro approaches to supplement each other for the production of candidate drugs. Then how to reduce toxicity level. The right combination of multiple properties of the molecule like exposure, activity, toxicity relationships are carried out. The adverse effects can be reduced by removing chemical groups causing toxicity from the drug design, then developing in vitro and in vivo system to identify potential toxic metabolites, then identifying chemical functional groups that have either minimal toxicity or no toxicity at all and are metabolically stable. And how to improve drug safety. Drug safety is the foremost consideration for producing a successful drug. The safety profile in a drug can be introduced if the following are addressed. The drug does not have the potential to form reactive metabolites. Is the drug capable of binding with microsomal fractions of proteins? Formation of reactive metabolites should be probed in tissues such as liver, blood, and skin. Whether the reactive intermediates formed are able to penetrate other organs to produce adverse effects. Identify proteins that are modified due to drug administration. Then identify the drugs that are affected. Are there any drug-drug interactions? When reactive intermediates are formed, does glutathione GSH level show depletion? Then many tools are available to achieve safety level of a drug. These include experimental as well as computational approaches. The experimental in vitro testing is done to find out whether significant protein binding occurs and what the chemical basis of such binding is, so that chemical binders may be eliminated from the drug design. Then computational approaches. The computational approach encompasses in silico methods to identify potentially toxic chemical groups in the early stages, for which several programs are commercially available which specialize in the prediction of mutagenicity and cardiogenicity. The advantages of in silico tool are as follows. The ability to screen more compounds in less time to accelerate the drug discovery process. The ability to run ADMET models on virtual compounds so that an appropriate compound may be chosen from the compound libraries for chemical synthesis. An understanding of the relationship between ADMET properties and the structure and physiochemical properties of compounds may be obtained. Tomorrow we'll be learning about the molecular mechanics and molecular dynamics. Okay, so thank you for listening.